Okay. Before we get into things, I would like to welcome chili pepper number 2. That's right, I got another. So this one is apparently way spicier than the hot banana. That's the name of the other one. This one is a cayenne. I don't know how you pronounce it, but it's a long red chili pepper. It's apparently, again, a lot spicier. I think it's 3 to 5 times spicier than the one which I got. I was gonna go and buy this orange one, which had like a Spanish name, but I decided to get this one because according to like flavor profiles, it has a bit more of like a smoky taste to it, which seems kind of cool. It says that you can harvest them as early as July. So this will be my revision plant. This will be my plant which will carry me through the next 3 months. But yeah. Let's crack on. Let's get it poppin', penny pinching. I don't know. Look, this is what we did yesterday. So I'm going to quickly go over it again, and then we'll erase it, and we'll get on with mass transporting animals. So, number 1, you have your gas exchange in insects. So the way this works is very simple. Gas enters through these tiny pores on the exoskeleton of the insect called spiracles. From there, it goes into the tracheae, which is like a small little, thin tube. And then from there, the tracheae branches off into tracheoles, as you can see by this diagram. The tracheoles have very thin walls, and every single cell in the insect's body is linked by a tracheole, meaning every cell in the body gets a good supply of O2. A short diffusion pathway. When we're talking about gas exchange, when we're talking about this topic, refer to diffusion. Diffusion. You refer to diffusion, you will get marks. It's easy as. So, there are 3 ways in order for insects to exchange gas. The easiest one is simple diffusion. Literally, it goes from an area of high to low concentration gradients via diffusion. That's literally it. Oxygen is from the atmosphere, it goes in and into the cells. Easy. Number 2 is abdominal pumping. Easy way to remember this is literally that it just pumps in and out. It draws in oxygen and it pumps out CO2 by pressure changes. If it's out here, there's a lot less pressure, so it draws in things. When it's there, there's a lot more pressure, so it draws out things. Easy. Abdominal pumping. And that occurs during intense exercise, typically when it needs to respire quickly. And finally, during anaerobic respiration. So, here's a rundown. During anaerobic respiration, you need to respire quickly, so you perform anaerobic respiration. A byproduct of anaerobic respiration is lactic acid. Lactic acid is soluble, which means it will affect the water potential of cells, making cells have a lower water potential. As a result of this, since the water potential is lower, it means that water from the tips of the tracheoles will move in by osmosis into the cells. This will create a pressure gradient because there is now less volume in the tracheoles. So, as a result, oxygen is very quickly drawn in by diffusion. Very easy. So that's insects. Number 2, gas exchange in fish. The mouth is the buccal cavity. That's where the water enters from. Water has less overall oxygen in it than air, obviously. So, they need to be able to do this very effectively, and they do. The water enters, and then it goes to one of 4 gill arches. In these gill arches, you have gill filaments, which are little stacks to increase the surface area to volume ratio. So these gill filaments then have gill lamellae. Gill lamellae just have thin walls and a network of capillaries. Each lamellae has a little network of capillaries, meaning there is a constant supply of blood and there's a constant diffusion pathway. Final thing you should know is that there is this blood supply provided by the capillaries is in a counter current flow. So what that means is water is going one way and then the blood is going another way. This means that it never reaches an equilibrium, and it means that oxygen-rich water is able to easily diffuse into oxygen-poor blood, and vice versa. So it never reaches this equilibrium because it's a counter-current flow. It's going different ways. And then finally, in humans, you have your lungs. Obviously, you have your trachea, which then branches into a bronchus, your bronchioles, your alveoli, which are your tiny air sacs. The alveoli have many adaptations. They have a increased surface area due to, I guess, the foldings. Surface area to volume ratio, very big topic, but you need to know well. They are surrounded by capillaries, which means oxygen is easily able to go into the bloodstream very, very easily. It's moist in order to absorb even more oxygen. Is it kind of... I don't have a word. I've forgotten the word. As the oxygen is kind of, I guess, intaken or absorbed by the water. You have your inspiration and expiration. So basically, in inspiration, your diaphragm flattens, or there's a better word for flatten. There's a better word for flatten, but I've completely forgotten. I don't know. Your diaphragm flattens, and your external intercostal muscles will contract. In other words, this will increase the volume in your thorax, and as a result, there is a pressure gradient because of this increased volume, there's less pressure. So as a result, air will diffuse in down this pressure gradient, and vice versa. The exact same thing happens in expiration. Your diaphragm does the opposite; it relaxes. Your external intercostal muscles contract slightly. The pressure gradient means that oxygen, or I guess in this case CO2, will be drawn out. So oxygen drawn in, CO2 drawn out. It's basically like abdominal pumping, but, a little bit more complex. Finally, surface area to volume ratio. The smaller organism, the bigger its surface area to volume ratio. In order for us to stay alive, because the diffusion pathway is too large, we need to rely on more complex and larger exchange substances, exchange surfaces, such as, the heart, the lungs, etc., etc. And we can maintain heat because of a smaller surface area to volume ratio, but other organisms, which are, for example, unicellular, can lose heat very efficiently, easily. Organisms in colder temperatures need to maintain heat, so they'll typically have a smaller surface area to volume ratio, like curved features, like a curved head. Meanwhile, in deserts and warmer environments where they need to lose heat, they will likely have a bigger surface area to volume ratio, such as pointed features, like pointed head, pointed ears. Just remember foxes for that example. Final thing which I just forgot about, but I read it there. Pleural fluid. That it basically is just a lubricant on your lungs to reduce friction. There you go. Entire topic in 9 minutes. Right. Woo! And what I'm going to do is for the next 2 or 3 days, when I do revision, I'm going to check how much I can remember from this topic to make sure I remember it. So, today we're doing mass transport in animals, aka the heart. That's literally the topic we're doing. I would argue it's a somewhat complex topic, but only because you need to know a lot of words. So let's see how much I can remember with barely any revision. I have not revised this topic once, so let me grab a drink. Well, let me just see. Okay, so So, we're gonna draw the heart as literally like, I don't know, I'm gonna draw it as a box, and this will make sense why in a second. Because there is kind of a gimmick here, so... So, first and foremost, the structures of the heart. Your atriums, your atria, and your ventricles. Very, very cool. You have your AV valve, your ventricular, what one of those? I don't know. I'm bad at speaking. Valves up there, and then this is your semilunar valve. Okay, now we've gotten that over and done with. There are 4 main blood vessels. Okay, let me see if I can remember this first time. You have, firstly, let's draw the lungs here. Oh god, how do I draw the lungs? I forgot. I just draw them as like small, like, there. Okay, these are your lungs. And you have your pulmonary circuit. So the way the pulmonary circuit works is it's your pulmonary vein and your pulmonary artery. So the vein, oh my god, how have I forgotten this? The artery is for oxygenated blood. So obviously your pulmonary artery is gonna go there. Your pulmonary vein so basically the pulmonary vein will take the, oh my god, hold on, how have I forgotten this already? Pulmonary vein into the lungs, and then your pulmonary artery goes back into the heart. No, hold on. No. I've fallen for the oldest trick in the book. The pulmonaries are the opposite of what they do. So veins are for deoxygenated, except for the pulmonary vein. And arteries are for oxygenated, except for pulmonary artery. So your pulmonary circuit, so that will be your pulmonary vein, that will be your pulmonary artery. Yeah, because it's going... Okay, I think that's right. I really hope that I've got that right, because that's like basic knowledge, really. So your first circuit is a pulmonary circuit. So you have your pulmonary artery, goes into the lungs, takes in oxygen, and then your pulmonary vein takes that oxygen back to the heart, where it then will go through the systemic circuit. And this is where you have your other organs. And you have your aorta. I'm just going to draw it like this. And then your vena cava. I feel like I've done this in the wrong order. Have I done this? Have I actually messed up? Have I done it in the wrong order? Are veins for oxygen? No. They go into the heart. Arteries go out. Let me check. Your lungs go to pulmonary vein. Okay, well I got the order, right? Pulmonary artery and vena cava is for deoxygenated. Yeah. Okay, you know what, mate? We should simplify this a lot further. Because clearly, doing this on an annoying diagram is not helping. Let's see. Okay. Here. In fact, we could draw this so simple. I've just realised. Okay. Your pulmonary vein. So we're going to draw the heart as a literal heart because symbolism. Okay, look. Look. Okay. So, from the heart, you have your pulmonary vein, which goes to the lungs. Where's my black pen? Am I being stupid? No. Okay. So from the heart, the pulmonary vein goes into the lungs. Okay. Where then the pulmonary artery goes back into the heart. And that's the pulmonary circuit. Your systemic circuit is where you go from the heart through the aorta into, I'm just going to draw like a person, to other parts of the body. And then from there, your vena cava takes it back to the heart. There you go. And I guess we should specify as well. So, that's the overview of the circuit. Your pulmonary, heart, lungs, heart, systemic, heart, body, heart. Easy. Ezra Collective. Just, okay, thank you. I don't need to know that right now, though. Okay. That's all good. Oh, it's warm all of a sudden. That's all good, right? That's easy. Yeah, I did mess up because I'm silly, but that was easy enough. Right. Let's draw little indicators, so. Hold on, is it the pulmonary vein? Ah, it's a pulmonary artery. So sorry guys, I got that mixed up. You know why? It's because these blood vessels do a lot, and it's kind of complex. So, there you go. Deoxygenated, oxygenated, oxygenated, deoxygenated. There you go. That's the systemic circuit and the pulmonary circuit. Nice and easy. Okay. Now, the 3 types of blood vessel. Your vein, Okay. Vein. Artery. Capillary. First of all, arteries take oxygenated blood. Okay. Veins. Yeah, obviously, because veins go into the heart. That was why I missed a mistake on it. Okay. Veins take deoxygenated. Okay, now we've gotten that cleared up. Let's look at these quickly. Veins have a wider lumen and pocket valves. They are aided by muscles contracting in order to go back up into the heart. So vena. Vena cava uses muscle contractions to go to the heart. That's basically all you need to know about the vein. The arteries have a narrower lumen, more elastic and muscle tissue. Because they need to withstand a higher pressure. Capillaries are 1 cell thick. And that's kind of all I really recall. Obviously for a short diffusion pathway. But I don't recall anything else about capillaries just yet. There are stuff you need to know about it in relation to the lymph, but we'll get onto that in a sec. So there you go. That's my wholesome little diagram about the veins, the arteries, the capillaries. I'll just kind of draw this to separate them. Okay. Okay. So, hemoglobin. And at the end of the topic, at the end of this stream, I will be doing some exam questions. So that's how we'll test if we've made it stick today instead of using keeping a mind map up. Hemoglobin. So red blood cells. First and foremost. I should have drawn it in red. That probably would have helped illustrate the fact that it was a red blood cell, but hey. Red blood cell. Okay. First of all, red blood cells have a flat, biconcave shape. This basically just increases the surface area. And they also have no nucleus, which allows for even more oxygen to be stored. So, the red blood cell contains a pigment or contains hemoglobin. That's my best drawing of it. Contains hemoglobin, which is a quaternary protein. Okay. And it contains iron ions in the heme group. So in the heme group, each heme group can absorb, I'm pretty sure, 2 oxygen atoms or 1 or 2 molecule. Alright, So, what else is there to know? The whole idea of actually how this becomes... Oh, I forgot the word. Disassociated, associated. It associates with oxygen. And this can be increased by CO2. Because CO2 will dissolve into carbonic acid, which increases the pH, causing a slight change in tertiary structure. That's called the Bohr effect. You also have the idea of a conformational shift. Basically, to begin with, it is actually rather difficult to absorb the first molecule of oxygen, but after it does, it will undergo a conformational shift, which makes it easier for the next two molecules of oxygen to be absorbed, but then that fourth one is a little more difficult to absorb because 3 out of 4 are already saturated with oxygen. 3 out of 4 have already been associated with oxygen. It disassociates with oxygen, obviously by kind of a Bohr effect, causing it to undergo... Carbon dioxide will cause a change in structure which causes it to disassociate with oxygen, but I'm not sure if there's another reason. So that's all the knowledge I can think of, so let's go check our book. Each polypeptide has a heme group with Fa2. So yeah, I was right, except 2. Not Fa plus. Okay. Red blood cells, biconcave shape. So yeah, a large surface area to volume ratio. I think when talking specifically about red blood cells, mention the ratio. They are flat for a short diffusion pathway. And they have no nucleus, which means there is more hemoglobin for more O2. I didn't mention that, but I'll just draw it now. A globular protein, in case you didn't infer it from the name. Hemoglobin's affinity... There you go, that's the fucking word. It's not difficult, it's affinity. Its affinity can be increased by the partial pressure of oxygen. So, higher partial pressure, the saturation of hemoglobin, and the partial pressure of CO2. And as we said, the saturation of the hemoglobin, how saturated it is. So, partial pressure is just the concentration of a gas in a mixture. We know that. Saturation increases as more binding sites are occupied. So the higher concentration of oxygen... Okay. So... it disassociates to release oxygen. A high oxygen saturation means binding sites are occupied in full. The higher partial pressure, the greater affinity. So, for this topic, your keywords are affinity, partial oxygen pressure, respiration, saturation, and association. And polypeptide, maybe. So unsaturated means no oxygen to bind to it. The level of saturation changes, so... Yeah, cooperative binding. That's it. That's what it's called. Co-op binding. So what that means is once the first binding site is occupied, remember it's called a binding site too. Binding site. So once the first binding site is occupied, it makes it easier for the second and third as it undergoes a conformational shift. For the fourth site, the chances of the relevant binding site is lower. So... there was one more thing, which was the Bohr effect. And the idea there is, in oxygen-depleted areas, there is a stronger affinity for oxygen. More CO2 means more oxygen release. Because carbonic acid causes a slight change in the tertiary structure. It decreases... Okay, here we go. So there's a lesser affinity to oxygen, which means it dissociates more readily. Okay, if that looks like a shit ton of stuff, that's because it is. But in reality, this can be simplified a lot, I think. Red blood cells, large surface area to volume ratio, short diffusion pathway. That's it. That's all you need to know about that. Hemoglobin, quaternary protein with a heme group, which acts as the binding site. Made up of 4 polypeptides. That's all you need to know. It associates with oxygen. It'll have a higher affinity to oxygen if there's a more oxygen or a higher partial pressure. More CO2 means more respiration, which means it's likely to have a higher affinity. During respiration, carbonic acid is created due to the carbon dioxide. This is called the Bohr effect because there will be a slight structural change in the polypeptide. This lessens its affinity to oxygen, meaning it dissociates more readily. Cooperative binding is literally the idea that the first molecule is a little difficult to bind, but then the second and third one can be readily associated more. They have a higher affinity, and then the fourth one, the chances of oxygen interacting with the relevant binding site is a lot lower. There you go. So, okay, we're on 34 minutes. Just gonna... Okay. So, one thing which I'm actually currently writing like a Google document just to like share this to everyone because I think it's very important and not enough people kind of acknowledge it. It's the idea of using keywords a lot. Because every topic has like 5 or 10 keywords, which if you put in your 4 to 6 marker, the writing questions, if you put keywords in your writing questions, guarantees you can get marks. I mean, I was doing a past paper the other day, I lost out on 2 marks, literally just for forgetting to mention surface area to volume ratio and diffusion. 2 easy words, which anyone knows, but I just simply forgot to mention them. And all it's needed is a quick reference to it, maybe a tiny explanation, and you can get marks. So for this topic, the whole topic of mass transport, in animals. Here's some keywords which I think are very good. Okay, we'll start with hemoglobin first. Affinity, binding site, saturation, association, and dissociation. The Bohr effect, cooperative binding, and partial oxygen pressure. Those words right there, that's your bread and butter for the hemoglobin topic. Let's say we got, very quickly, example, let's say we got a 6-mark question talking about, I don't know what it would ask in a particular context, but let's say it was on about, describe how hemoglobin allows for oxygen to be delivered to respiring cells. You can then talk about how oxygen will bind to the binding site because it has an affinity for it. It will associate. You could talk about how it's cooperative binding. You could talk about how if partial oxygen pressure was increased, it would mean that there are more oxygen molecules and thus a higher amount of, or a higher chance of association or something like that. Oh, great, okay, Portal 1 with the RZA. I rip shit hardcore like porno flick bitches. Classic like turntables, beat flops, or floppy diskettes. A young Tom Cruise is with me and it's risky business. I deliver more gifts to the game than 1,000 Christmases. That one? So that's your bread and butter for this topic. Hemoglobin, red blood cells. To be honest with you, I don't know if we'll get a 6-mark or even a 4-mark question on red blood cells. It seems uncommon. It seems unlikely, to be honest. It seems unlikely that we would get like a massive question, especially because that's a lot to remember. But that is a lot of words to remember. Affinity, association, binding site, the Bohr effect. A, B, B, C, P. AABBCP. I guess if you guys really want an acronym for that, then affinity, association, binding site, Bohr effect, co-op binding, partial pressure. If you really want an acronym, AA, double B, double C. In terms of the heart and like mass transport in general, literally just the veins, like the blood vessels. I always call it veins because I'm used to just saying that, but talking about literally just naming the blood vessels, that's easy enough to do. Valves, your pocket valves and your actual valves. Your left ventricle muscles are thicker because it needs to withstand a higher pressure, provided because in your left ventricle, that is where it's sent, further. It's sent around the whole body, not just to the lungs. Valves prevent backflow of blood. We got that. AV valves are for the ventricles to the atriums, and your semilunar valves are for the arteries to the ventricles. Your right atrium has pacemaker, Jesus Christ. Pacemaker cells. And it's your left ventricle has a thicker side. Your right atrium has your pacemaker cells. So yeah. Okay. Let's quickly go over diastole and systole and all of that shit. Okay. very quickly, this should be easy because I've literally just gone over this. So let's try and remember. Diastole. Atrial systole. And atrial diastole, or I think it's called ventricular. I always call it ventric, but I think it's ventricular. Is it diastole or systole? Yeah. So this one's very easy. Diastole, systole, systole. Okay. A good way to remember what these are, in case you genuinely have no idea, is systole. I always spell it, purely by error, as systole with a C. And C, the same first letter as contracting. That's what it does. In this sense, your atrium will contract. In this sense, your ventricle will contract. So firstly, diastole is relaxing, okay? So cardiac relaxing, cardiac diastole. So your cardiac muscles are relaxed. Okay? Blood will enter into the atrium and possibly into the ventricle. Okay? Okay. At this point, your AV valve is open. Your semilunar valve is closed. Next, in your atrial systole, the atrium will contract, which creates a pressure gradient. This creates a pressure gradient, and it will diffuse. I'm going to turn off chat so you guys don't see. A pressure gradient, so the blood will move into the ventricle. Okay? Then, at that point, your atrioventricular valve will snap shut. Basically the ventricle will firstly contract, which creates a massive pressure, a massive pressure gradient. Your AV valve will snap shut, as your semilunar valve will be forced open due to the amount of blood coming through. And I'm pretty sure that your atrium will contract or something as well in order to push it through. Let me just try remember. So, during cardiac diastole, all chambers in the heart are relaxed. So I should have put chambers there instead of muscles. So blood will enter the atrias through both veins. The atrioventricular valves are open, which allows blood to enter the ventricles, but the semilunar valves are closed, like we said. In atriosystole, the pressure in the atria, the atria contract, significantly increasing pressure in the atria because there's less volume, causing the blood to be forced into the ventricles, down the pressure gradient. Finally, in ventricular systole, the ventricles contract, beginning from the apex. I forgot that, the apex. The very bottom. It starts at the apex to force out blood, forcing out the blood to leave through the arteries as the semilunar valves are pushed open by blood due to the high pressure. The pressure in the arteries causes the semilunar valves to snap shut as the AV valve opens again to allow blood to enter, preventing backflow. Yikes, that is a massive topic, to be honest. That's a lot of shite to talk about. So during cardiac diastole, all chambers of the heart are relaxed. Blood will flow in to the atrium and the ventricles. Via gravity, I guess, would be your gimmick there. Your atrioventricular valves open, allowing blood to pass through. Next, the atria will contract. This obviously reduces the volume in the atriums and causes a pressure gradient. So once all the blood is forced into the ventricles, that's where the ventricles will contract. The AV valve will snap shut to prevent backflow as it's forced from the apex, it's forced into, obviously, the arteries out of the semilunar valve, which is forced open. Massive topic, and we're still not done. We have one more thing to talk about, and that's the lymph. The whole gimmick with the lymph Oh wait, hold on. There's still a bit about blood vessels we need to do. Endothelium, that's like the slightly smaller layer. I forgot about that. Oops. Capillaries have pores in the endothelium to allow substances to diffuse. Arteries can contract to restrict blood flow. There you go. What do you call them? Your arterioles, your atrioles, and your venules. I don't know how you pronounce these. I don't remember how you're supposed to pronounce these. In other words, they're mini veins. They have mini valves. They bring deoxygenated blood from capillaries to the veins. So here's an easy way to remember. Artery into your atrioles, into your capillaries, into your venules, I'm just going to call them, into your veins. That's kind of a capillary network. Okay, then your lymph is somewhat simple to understand, to be honest. You have your capillary. Oh, what time is it? That was weird. Okay. Your capillary. There is a high hydrostatic pressure in the blood, which is caused due to the fact that the arteries contract, something like that. Blood in the capillaries is at a high pressure as it is coming through the arterial end, creating a pressure gradient between the blood. Okay, so high hydrostatic pressure is coming through. Okay, so there's a high hydrostatic pressure. This creates a pressure gradient between the tissue fluid and the blood, which causes, molecules such as oxygen and I don't know what fuck's sake. Just drew on my thing. I don't know what molecules, to be honest. She didn't really explain. This forces, okay, I guess literally small molecules. This forces small molecules to go from the blood to the tissue fluid via diffusion out of these tiny little pores. Because there is now a higher concentration of molecules in the tissue fluid than the cells, molecules enter the cells by diffusion or active transport. This forces out waste products which enter the lymph, which is eventually cycled back around to the capillaries. While that sounds complex, I kind of have it in my head that it's quite easy. So let's finally, after almost an hour of revision, let's take a minute. Let's take a minute. And then we'll go do some exam questions, which we are being forced to do, so I guess it's not really my choice. Right. Explain the advantage of the change in dissociation curve for hemoglobin during vigorous exercise. Okay. During vigorous exercise, there will be more cells will need to respire more. This causes more CO2 to be present, which reduces the pH, or increases the pH. Reduces. This reduces the pH, causing a slight change in the tertiary structure of hemoglobin. Hemoglobin, as a result, will dissociate more readily with oxygen, meaning there is a quicker supply of oxygen. List 3 reasons why a large multicellular animal, such as a mammal, needs a transport system. Okay. I will list 3. Number 1, low surface area to volume ratio. That's the most obvious one. A low surface area to volume ratio means a higher diffusion pathway. So complex oh wait, hold on. A greater need for O2. There you go. Low surface area to volume ratio, higher diffusion pathway, greater need for oxygen. Is that correct? I don't know. Let's find out in a minute. Fish have a single closed circulatory system. State the meaning of the terms single circulatory system and closed. Single means it only blood passes through the heart once in a circuit. Blood travels through a singular circuit. I don't know what else to put for that. Maybe we can get a definition though. Closed circulatory system. Obviously, an open circulatory system like an axolotl means molecules can't just enter and exit. So, I don't know. They travel through veins. Describe and explain 4 ways in which the structure of a capillary adapts it for the exchange of substances. Okay. 4 ways in which the structure of a capillary adapts it. Okay, 1 cell thick for a short diffusion pathway. Pores in endothelium allow for small molecules to exit via diffusion. High hydrostatic pressure allows for molecules in blood to exchange with tissues. I can't think of another one. 1 cell thick, pores in endothelium, high hydrostatic pressure. Maybe, I guess I could elaborate as blood comes from arterial. I can't think of a 4th. Figure 5 represents a transverse section of an artery and vein. State 2 other ways in which the wall of an artery is different to the wall of a vein. Narrower lumen and more muscle / elastic tissue. Easy. Blood in the arteries has a high hydrostatic pressure. Hydrostatic pressure is generated in the heart. See, I don't fully recall this. High pressure I don't know why the hydrostatic pressure is formed. High pressure from arterial end of capillaries due to atrium contracting. Explain why hydrostatic pressure of the blood drops as blood moves away from the heart. Because there's literally... How the fuck are you meant to explain that? Explain why hydrostatic pressure drops as blood moves away. Because there's less pressure. I hate questions like that, where it literally gets you to state the obvious, but in a complex way. I don't know. Why does hydrostatic pressure reduce as it moves away from the heart? Because there's less force applied by the heart. I don't fucking know. Literally no fucking idea for that one. Explain why a vein may be described as an organ, because it's comprised of multiple different types of tissue. It is comprised of many types of tissue. Use information in the table to suggest the thickness of a capillary wall. 1 cell thick. Suggests a thickness, maybe 1 as it needs a small diffusion pathway. The diameter of the artery was 4 millimeters. Calculate the diameter of the lumen of this artery. I'm not going to do that because I don't have a calculator. Explain how the elastic tissue in the wall helps even out the pressure of blood flowing. Allows artery to contract / relax to regulate pressure of blood. I don't know whether that's enough, but fuck off. Let's go check the mark scheme. I did a lot there. I wrote a lot. Right. Just go check chat. Okay, Luke's put nothing. Right, good. Do I even have a mark scheme? I'm willing to bet I don't, do I? I'm willing to bet I don't have the mark scheme. Where could I have put the mark scheme for this? We might have to just end things. I might have to tell the teacher that I was not given a mark scheme, even though I definitely was. I definitely was given a mark scheme for this. I definitely was. I just can't figure out whether it was. Anyways, I think that's enough revision for one day. I'm knackered, to be honest, and I still have stuff to do. I have to go tidy up my, what do you call it? I have to tidy up my folder, because of course our teacher said, go tidy up your folder. What time is it? You know what I'm going to do? I'm going to do Creed Rise to Glory boxing. I am the most unfit person alive. I might as well spend like 20 minutes punching. I'll catch you guys in a bit.